Hello and welcome everyone, and thank you to Anshul and Gabrielle for organizing the session and inviting me to speak. I'm Adrian Sanborn, and I'm excited to tell you today about our work combining deep learning and high throughput experiments to understand the sequence determinants and function of transcription factor activation domains. So I'll be talking mostly about transcription factors, or TFs, and these are proteins that bind to DNA and affect transcription of genes in some way. These TFs are central in all sorts of pathways involving signaling a disease. They perform the last step in many signal transduction pathway, where a small molecule signal or protein signal gets turned into a change in gene regulation. And because of this, TFs also define the identity of different cell types, and mutations involving TFs drive many cancers and other diseases. And these TFs are also modulated directly by the binding of small molecule hormones and drugs. So it's really critical to understand the function of these proteins. A typical TF has two essential parts. There is a DNA binding domain that determines the kinds of DNA sequences in the genome where the protein binds. And this can often be right next to a gene in the promoter, or it can be quite far away from the gene in an enhancer sequence that still regulates that same gene. And then the TF also has a second domain called the effector domain, which is the domain that has some effect on transcription. So it could recruit other proteins involved in regulation, or it could directly modify the DNA of the chromatin. So I'll be focusing on activation domains, or ADs. And these are effector domains that specifically increase transcription of a gene. And it does so often by recruiting complexes centrally involved in transcription. So one of the most interesting things about activation domains is that they have very highly conserved function, but not sequence. So it's been known for a while that these ADs are highly diverse in sequence. And some screens of random protein sequences, in fact, found between 1% and 4% of random protein sequences can spontaneously form activation domains. And what that means is that, unlike DNA binding domains, these activation domains have not been systematically identified. So with DNA binding domains, you can study like a canonical example of a DNA binding domain and then extrapolate to many other homologous sequences. But because there aren't homologous sequences for activation domains, we can't do this. Despite the high sequence diversity, many of these activation domains actually retain activity even when transferred between yeast and human. So you can take an activation domain from a yeast protein and put it into human cells, and it will still activate genes. And so my work here is focused on these two essential questions. First, what are the essential features of activation domains? So it's not obvious what's being conserved, but something about the sequence is being conserved, which allows it to keep its function between yeast and human. And second, what is the mechanism that underlies this activation that enables this kind of sequence plasticity? And because we're talking about a large, diverse set of sequences, we decided to study this question by systematically characterizing the function and mechanism of all the activation domains in the yeast genome. And so I'll tell you about sort of two essential parts today. First is a screen that we did to measure and detect these activation domains in yeast. And second, a deep learning algorithm we trained called Paddle that allows us to actually predict the strength of activation given just the protein sequence alone. So how do you actually find activation domains and measure activation? So this is the classic reporter activation assay, where we take an artificial transcription factor, which has a DNA binding domain, which brings this transcription factor upstream of a GFP reporter gene. And this is the signal that the activation we're reading out. And then we take different fragments from transcription factors and clone them in here to this purple segment and ask for different TF fragments, which ones activate this GFP gene. And then we can track the expression of this using an mCherry reporter as well. So to do a pooled screen to discover all the activation domains in yeast, we actually designed a pooled tiling library where we have DNA tiles encoding 53 amino acid fragments of these TFs that cover all 164 yeast transcription factors. And then we generated a pooled library of plasmids that we could then introduce into yeast cells in such a way that each yeast gets exactly one plasmid. And then we measure the activation using fluorescence activated cell sorting. So specifically, we first sort the cells based on their expression level of the artificial transcription factor using mCherry. And then we sort the cells into 8 different bins using their GFP signal to read out how much activation occurred in each cell. And then by sequencing the cells that fell into each of these 8 bins, we can see for each individual protein tile how much activation that tile caused. So for example, the teal tile A, most of the cells fell into this bin number 2, indicating no activation relative to negative controls. Whereas for tile B, most of the cells fell into this bin number 7 with very high GFP signal, indicating roughly 100-fold activation of GFP relative to negative controls. So in this way, we obtain measurements of fold activation of GFP for each protein tile. And then when we plot these tiles by the position on the protein, we find that the activating tiles cluster into discrete activation domains. So here the x-axis is showing position on the protein for 3 different proteins, and the y-axis is showing fold activation. And these tiles here are clustering into discrete activation domains, reproducing known activation domains from these proteins. And to give you a sense of the data, this is activation for 60 different transcription factors, where the positional activation is colored darker for high activation, and the activation domains that we've annotated systematically are boxed in green. And so in total, we find across 164 proteins, we annotated 150 activation domains. And in fact, by doing some simple sequence analysis of the protein sequences of these activation domains, we find that these ADs do share no common motifs. What they do share is an enrichment of hydrophobic and acidic tiles. So when we binned the tiles based on their total hydrophobic content and their net charge, we found that the ones in the lower right corner activated the strongest. Now, hydrophobic residues were known for a long time to be necessary for this kind of activation domain, but there's been conflicting evidence for how important acidic residues are for activation. So to more systematically answer this question, we also built in a series of mutant sequences into the library. Specifically, we took a few known activation domains and systematically mutated away either aromatic residues, so highly hydrophobic residues, or acidic residues. In this way, we can titrate systematically the amount of hydrophobicity or the amount of negative charge. And what we found when we plotted the activation as a function of the hydrophobicity or the net charge is, first of all, a systematic drop-off in the activation as a function of the number of mutations that occurred. But secondly, the activation was mostly dependent on the number of residues mutated, not their position, indicating that somehow sort of the total hydrophobic content or the net charge was the most important feature, rather than specific key residues for activation. And this property was consistent across all 8 activation domains that we tested in this way. So then we wondered, can we just predict activation just based on amino acid composition? And to test this, we trained a simple neural network, a fully connected neural network, to predict activation strength in the form of z-scores. And this network was given only amino acid composition. And we split our train valid test data by protein because adjacent tiles have overlapping information. So we wanted to make sure those were all put into the same split. And in fact, this network performed quite well, just given amino acid composition with an R-squared of 0.66. And this is showing the performance on test data. But we also knew that this couldn't be the full story because we had also experimentally tested scrambled sequences for these known activation domains. And that let us measure experimentally the importance of amino acid composition on its own versus sequence. And what we found was that for each of these 8 activation domains and for each of the 8 scrambles, the scrambled sequences, some of them activated very strongly, as strong or even stronger than the wild type sequence, but some of them activated not at all. And so obviously these sequences all have the same amino acid composition within the scrambles, and therefore the neural network that predicts just based on amino acids composition could not possibly predict these differences. And so we know therefore that both amino acid composition and sequence contribute to activation. So then we trained a more complex model based on sequence to predict activation. And specifically, we use a convolutional neural network. And this was done together with Ben Ye, a really fantastic master's student in the computer science department at Stanford. The architecture of this is as follows. We have convolutional layers that have a window size of about 10 amino acids. In total, 9 convolutional layers. We do maximum pooling along the sequence at the end, and then there's 2 fully connected layers that give us the output prediction. And we called this network PADDLE, which stands for Predictor of Activation Domains using deep learning in eukaryotes. So PADDLE was able to perform significantly better than the simple amino acid composition-based neural network. So here's an example of the ARG81 protein with the experimentally measured activation shown along the y-axis. And the predictions from PADDLE not only reproduce the position of these activation domains, but also the relative strengths of activation. And this is the performance across all the test protein sequences. The significant improvement in performance is measured by the R-squared score, up to 81% of explained variance. And PADDLE is not only able to predict the position and strength of wild type sequences, but it can also predict the effect of mutations. So we tested it on these scrambled sequences, which are a difficult prediction task because they have very similar amino acid composition. And PADDLE is able to distinguish which one's activating which ones don't. And we also took a single known activation domain and tested a whole bunch of different mutant sequences and homologs of it And Paddle was able to very accurately predict the effect of these mutations on this known activation domain. Just a few notes about the optimization of Paddle I thought would be interesting to share with this audience. We did find that these deep convolutional neural networks perform better than shallow ones, suggesting that model complexity was an important factor. Predictions improved slightly when we included predicted secondary structure and predicted disorder as input compared to just predicting on sequence. So this was used in the final model as well. To get the best performance, we averaged across an ensemble of 10 different models trained on different trained val splits. We also mitigated overfitting by using early stopping. We tracked the validation loss and stopped when it was looking like it wasn't improving. And finally, we tried a few data augmentation strategies. For example, adding close homolog sequences and assuming that those homologs activated the same as the Saccharomyces cerevisiae sequence. But actually, adding this additional diversity didn't seem to improve the performance. So given that we were able to very accurately predict activation in yeast transcription factors, but given also that there's quite a good conservation of function between yeast and human transcription factors or activation domains, we wondered then if Paddle could predict activation domains in human proteins despite being trained only on yeast sequences. So we ran Paddle across a whole bunch of yeast human transcription factors and generated these predictions, and then picked 25 of these predicted activation domains at random so that they would be a representative sample, and tested them in a human luciferase assay. And we were surprised to find that of these 25 randomly chosen predicted activation domains, 23 of them activated significantly in human cells as well, which suggests that Paddle is in fact learning properties of activation domains that are conserved from yeast to human. And furthermore, this tells us that we can actually use Paddle to annotate hundreds of activation domains in human TFs and also virus TFs. And we've now used Paddle to generate predictions for TFs across many different species. And those are available online at paddle.stanford.edu. We then studied what Paddle was learning to try and better understand the mechanisms of activation domain sequences. So one thing we learned from studying Paddle, what Paddle was predicting, was that besides the role of hydrophobicity in enhancing activation, clusters of hydrophobic residue can also inhibit activation. So we noticed that Paddle was making predictions like this, where these shorter sequences inside the RDS1 protein were predicted to activate. But upon extending the sequence upstream and adding these bulky hydrophobic residues, Paddle then predicted that this region would no longer activate. So we systematically characterized this property by designing a second library to explore all of these lessons we were learning from Paddle predictions. And to study this systematically, we took a short known activation domain and put it next to a variable region of design sequences that have either very high or very low amounts of hydrophobicity or net charge. And in this way, we were able to generate this sort of phase diagram telling us how much this variable region either increases or decreases the activation of this short activation domain. And in fact, we found that very high levels of hydrophobicity and of course, positive charge significantly inhibited activation. We also used Paddle to identify the short key regions of activation domains, which we're calling core activation domains. So you can generate predictions for arbitrary subsequences of transcription factors. And when we do this, this is what it looks like with the length of the subsequence on the y-axis and the center position of the sequence on the x-axis. We see that there are these regions, these clustered regions that activate significantly or predicted to activate despite being relatively short. And so in the second library, we also did a shorter tiling measurement where we looked at 13 amino acid tiles and confirmed many of the predictions from Paddle that identified short 13 amino acid regions that could activate on their own. So in the second library, we tested a lot of mutants of these core activation domains, these 13 amino acid sequences, to try and understand the mechanism a little bit better. So first, we looked at alpha helix formation. Alpha helix formation was thought to be an important factor for an activation domain. And to address this question, we looked at the non-hydrophobic residues in the center of these core ADs and mutated each of them to either proline or to alanine. Now, proline would break a helix, whereas alanine would promote a helix. And so we found some activation domains that look like this. When we mutated to alanine, they still activated just fine, but when we mutated to proline, their activation dropped to background levels. And this tells us that the PUT3 core AD needs to form an alpha helix to activate. But for the PIP2 example, the core AD didn't seem to care whether its residues were mutated to alanine or to proline, and this tells us that the alpha helix is actually dispensable. And we found that only about 1/3 of the core ADs we looked at required an alpha helix. Next, we asked the same question again about amino acid composition, but now focused on these 13 amino acid core ADs. And so specifically for each of these 28 core ADs, we generated over 30 scrambled sequences and asked how they distributed relative to the wild type. And here, this is sorted by sort of the ratio of wild type to the scrambled sequences. So there are a few activation domains that couldn't be scrambled. When they were scrambled, they would no longer activate. But actually, the majority of the sequences could scramble just fine, and they would activate similarly to the wild type. And so we're thinking of these as amino acid composition-driven activation domains. And these on the right also were ones that tended to have more hydrophobicity. Now, there's also an interesting correspondence between these two properties I just described. So if you plot the proline inhibition on the y-axis and then the wild type to scramble ratio on the x-axis, we find that there's essentially these two classes of activation domains. The ones up here on the upper right are sequence defined, meaning you can't scramble them, and they also form an alpha helix. Whereas the majority, the ones down here on the lower left, actually are amino acid composition driven. You could scramble them and they'd still activate. And they also don't need to form an alpha helix and are probably disordered. So just to tie this all together, it's interesting to ask the question of why do these activation domains have such sequence diversity? I think there's several important advantages to this. First, such plasticity of the sequence allows new activation domains to readily evolve, and therefore new transcriptional circuits to evolve in response to the organism's needs. This also means that the strength of activation can be tuned gradually by changes in mutation. And third, these activation domains can simultaneously evolve binding sites to other proteins while being activation domains because of the plasticity of the sequence underlying. So actually what I've told you today is only a part of the full story. I don't have time to tell you about our efforts to do in vitro binding assays with mRNA display and also some systematic structural modeling of these interactions, but you can read all about that in this work that's now published. So this work has all been supported by my advisor, Roger Kornberg, and Ben Ye contributed substantially to the computational infrastructure, both for machine learning and for the design of these sequences. There are many others who contributed to this project listed here. Here are the funding sources. So thanks for your attention, and I'm happy to take questions now.